I will now introduce our next speaker, David God. He has over 30 years of experience in the toxicological risk assessment of other chemicals and regulated products, such as pharmaceuticals and cosmetics. He was a toxicologist at the Food Standards Agency in UK, and he is now training a new generation of toxicologists for the Food Standards Agency. David, the floor is yours.
Thank you, Chairman. Thank you. I am going to talk today, and I hope you don't get too disturbed. My next door neighbours are having their garden changed, and I can hear the machinery working. I hope it doesn't get picked up with the microphone. So I'm going to talk briefly about the outline of toxicological test requirements, the rationale for testing them, considerations around mutual acceptance, the use of traditional and tiered approaches, some thoughts on the use of old data and dealing with uncertainties, and finish with the crystal ball of Gypsy Rose got as I speculate on some other things we may need to think of in the future. Toxicology's got a long history. We can trace it back to classical times, and indeed De Materia Medica from Roman times was the basis of European pharmacopoeias through to the 19th century because of the timelessness of the empirical work based on trial and error. But really, toxicology can date back to the 1 minute it's not responding, then it's oversensitive, to the key concept put forward by Paracelsus in the 16th century. All things are poisonous, only the dose makes things non-poisonous. But of course, we had limited ways, and it was really the explosion of methodologies and analytical ability in the 20th century that made it forward. With the development of inbred strains of animals and the development and characterization of toxicological tests, key works such as the 1940s with Brody's work on methyl orange and R.T. Williams' publication of detoxification mechanisms. But it's worth bearing in mind that when we talk about these, the analytical sensitivity in the 1950s, when the US put the Delaney clause in their legislation, was tens to hundreds of parts per million. We're now talking about parts per quadrillion. And of course, for those of us who prefer books, 1975 and the publication of Toxicology, the Basic Science of Poisons by Cassaray and Doole. But regulatory toxicology is, as I say, relatively young. And really we can trace its formulation and perhaps its development to the 1960s, and one incident in particular. Thalidomide. For those who aren't familiar with it, thalidomide was a drug developed in the 1950s for the treatment of morning sickness during pregnancy, which had the unfortunate side effect that it was a teratogen and led to skeletal abnormalities in the children of mothers who had it. Of course, there was already a requirement to show drugs were safe at that time, but there wasn't a means of telling you what to do. And initially, all that was involved was convincing the reviewer. And European reviewers at that time were convinced by the data they had. And the US reviewer for the FDA, Francis Kelsey, wasn't convinced that they'd considered everything and held it up, which is why the US had less of a problem than Europe and Australasia with thalidomide. And following this, there was a codification of the required data. Authorities started specifying what was needed for pharmaceuticals, and this eventually expanded to other areas, such as pesticides and food additives, and was enhanced and evolved as new developments came along. So they subsequently drew up a list of required tests in areas on short and long-term toxicity, carcinogenicity, reproductive toxicity and teratogenicity. Genotoxicity followed later, of course, metabolism and kinetics. They were based on animal tests, often required 2 species of animals, a rodent and a non-rodent, or were required to be performed in a specified species. Food law, of course, dates back several centuries, but there was little progress till the 19th century, when it was estimated that up to 70% of the bread on sale was adulterated with things such as plaster, rat poison, you name it, they put it in. And really it was in the mid-50s when there was a recommendation that an expert committee should be formed to look at the administrative aspects of chemical additives and their safety, which led to the formation of the joint FAO-WHO Expert Committee on Food Additives, or JECFA. JECFA described the general principles of the first and the methods for risk assessment, which they published in their reports. But as time progressed, and in the 1980s, they recommended that this should be reviewed, which led to the publication of 2 environmental health criteria monographs on the safety assessment of food chemicals and pesticide residues, which were updated in the early 2000s and led to the most recent publication of EHC 240 in 2007, on the principles for the risk assessment of chemicals in food. We've seen the risk assessment paradigm in Taxis's talk this morning, so I won't go through it again. I apologize. There is a delay from me pressing the button to the slides changing. But the place where toxicological risk assessment really plays its major roles is in the hazard identification and hazard characterization as part of the risk assessment, which goes together to form risk characterization with this more important part of integrating the evidence. So if we turn to the test requirements, we'll talk about the traditional and now the more modern tiered approaches and putting those studies into context. And one question that many people ask is, why do we argue for guidance rather than mandatory requirements? Well, one of the simple reasons is that if it's mandatory, i.e. in legislation, it's fixed at a moment in time and very difficult to change, whereas guidance can keep up with adaptations to new scientific knowledge and can also provide the flexibility to allow things to be said to give the what's required without specifying how much what. For instance, in the food additives guidance, we talk of negligible absorption. What does that mean? Well, it depends on the chemical because it's a case-by-case judgment. For what some chemicals, 1% may be negligible. For others, 1/100 of 1% may be negligible. So the traditional guidance, and best example of that was pulled together by the Scientific Committee on Food for the EU, which really dated back to the 80s, 90s, and was still used in 2000, its latest revision, and was adopted by EFSA in the early days. Its emphasis was really on not missing things, identified the need for specific studies, and appeared, and I choose that word deliberately, to require them, but still required data from 2 species. So it set out the guidance for how the dossier should be described, set out requirements for other parts of data. You heard them talked about in the enzyme talk, so I won't go into them in detail, but it told people this data was required, but really nothing about why it was required. And then it sets a general framework for toxicological evaluation so that you could demonstrate the substance didn't pose any appreciable risk to humans, and thought about average and vulnerable consumers. Said that the study protocols would be international agreed protocols, the most up to date, but it allowed you to justify any different methods, and that protocols should be developed on a case by case. And the first of this guidance was written pre-GLP, so that it then adopted with the principles of GLP from a specified date. The core areas it required studies, in which you can see for other regulated products: metabolism, kinetics, subchronic toxicity, genetic toxicity, chronic and carcinogenicity, and reproductive toxicity. But it highlighted a number of other areas that maybe needed to be thought about. And it also included in that some areas that may be generated for other uses, such as acute toxicity or skin and eye irritation and sensitization. So it said that you should submit the range of studies recommended in the core set and should consider whether anything else would be required or might be appropriate. And now, to my mind, that's the most important statement. Toxicology and toxicological testing of a regulated product is not a case of going down a list of studies and just doing them. It is a case of looking at the evidence that comes from those studies and deciding what data you need to achieve the aim. And that is to protect, in most cases, human health. But it is said the reasons for these less than usual studies should be described scientifically. It wanted all results. We had some talk this morning about the new transparency regulation and the new one assessment, but we've always had a requirement for all results, favourable and unfavourable, to be submitted and discussed. And any other relevant data that you haven't produced but someone else has. Now, for a new chemical, this is unusual, but for older chemicals or for changes in use, information from the literature can be included and must be included. In practice, of course, it was always possible to justify not doing something or doing something different. And really, over time, using this guidance, a number of its seeming requirements were effectively discarded as they weren't necessary to make the decision that the advisors in be it the SCF or in the early days of the EFSA panels were providing. So some changes came, and around 2007, after EFSA 240, they started a revision of the food additives guidelines, and a new approach was taken. A tiered approach, which is really designed about thinking why you're doing the test, thinking what test you're doing, and pulling all the information together and integrating it. So that's the physicochemical data on the compound, information on related compounds, structure-activity relationships, and of course the need to bear in mind other influences on our requirements, such as the 3 R's of refinement, reduction, and replacement in animal welfare. And inherent in this concept is the rationale that results at higher tiers effectively supersede those at lower tiers, but that you don't need to go further in the tiers than is necessary to provide adequate assessment of the risks for humans. But also, it should, if applied correctly, strengthen the scientific basis for approach. It has a number of common elements, and they will apply to all categories of compound, and are specified in more detail in vertical guidance for each group of compounds. But essentially, it said this data, why was it needed? And it was important to realize its use in an integrated evaluation of the toxicological risks of the compound. So chemistry and specifications had always been required. And why? Because we need to know if there are things in there that need to be thought of when we're assessing the toxicological risk and need a toxicological evaluation. It may be that the levels set are already control that risk in the specification. But that's risk management, not risk assessment. The assessment is why it's there and what it is and what it does. It expanded the various areas for which of the identity, trying to provide more guidance on the more complex things like complex mixtures, botanical sources, all of which is part of the hazard identification step. Specifications and demonstrating that the material you produce and have tested is the same as put on the market. You've heard much of this in Christina's talk on enzymes assessments. The manufacturing process, again, does it introduce or produce any other compounds which may need to be controlled by risk managers in the material on the market? And evaluators were made clear, we need the detailed information. We may not need to describe that for the outside world, and this could be a challenge with the one assessment, but the evaluators need to know exactly what's there so they can use their judgment and skills to think, have you thought of everything correctly? Or if not, to come back with more questions to you. Stability again. Does the material go off over time, and if so, are more hazards generated? But we recognise you can't cover every eventuality with this, and it has to be based on the intended use. So if you're intending to use something only in cold products, providing data on high temperature heating of it would not necessarily be of value. We talked about the other approaches, and I'm aware of the time, so I'll move to the toxicological studies. And the tiered approach balances the data requirements against the risk. It initially uses less complex studies to obtain hazard data and dose response data, going to more complex systems and tests if they are required to assess the risk and to make decisions. So there's a minimal data set applicable to all compounds under Tier 1. Tier 2, which is more complex, comprehensive data, and Tier 3, which is really more for specialized uses or for the way we need to think about specific systems. So Tier 1 essentially comprised the initial guidance, and this has been adapted in other areas since, of an in vitro genotoxicity suite, which would trigger, if positive, in vivo genotoxicity, an absorption study, and an extended 90-day study, either of which could trigger Tier 2 studies for reproductive toxicity, chronic toxicity, and toxicokinetics. If absorption was not negligible, or if toxicity was observed in the 90-day study, even in the absence of absorption. Tier 2, apart from the in vivo genotoxicity, which really is semi-independent of the rest, because it's only triggered by if you have positive results in the in vitro suite and does not have any follow-up in Tier 3, comprises of single dose and absorption, distribution, metabolism, and excretion studies to provide evidence of whether or not there is accumulation, and then chronic and reproductive toxicity studies to decide if from their results whether there is a need for mechanistic or other data, or whether we have sufficient dose response information to complete the assessment. Tier 3 is really about doing detailed human toxicokinetic studies, mode of action studies to understand how and why the chemical works, or how it affects specific systems such as the endocrine system or developmental neurotoxicity. And it also recognizes there are some areas where currently, at the time we wrote it, there were no validated studies and a weight of evidence approach was needed, such as for allergy and intolerance. It called for other aspects because it was written at a time when the reevaluation program was starting and going, and had realized that there was a need for older compounds to get a systematic review of the literature and to have that described. And importantly, it requires the applicants to interpret the data and draw conclusions on the significant findings, not only in each study, but in the package overall, identifying the key critical endpoint and the point of departure for that endpoint. Of course, when submitting the information, it wanted the key studies and references, and critically, it stressed that the need for unpublished toxicity reports to be submitted in full, because summaries of unpublished reports are not acceptable. Those of us who have years of looking at the reports know that it is inevitable that you can sometimes see things in the collection of data, and we are talking of huge volumes of data in these reports done to regulatory requirements, that allow you to spot something that the lab conducting the study has either missed or that someone has ignored and not explained why they've ignored it. There is also a requirement for the exposure in two scenarios, one for something that's not being used anywhere, and the second for an extension to the use. And remember, these are theoretical estimates based on what the applicant wants that can be used in the risk assessment to compare with the hazard dose response data from the hazard characterization to see whether in the risk assessment characterization we need to say to the risk manager, you need to control this. Genotoxins will not be allowed in regulated products, but may be present in the presence of monomers in their process or residuals. And finally, some practical considerations. Is it done to an OECD guideline or equivalent? When? Why? What was it? Was it the current date? Has it been done to GLP? Is there a signed compliance statement? Is it from an accredited countries? And if you can answer those questions positively, congratulations. It's mad. And that's not mutually assured destruction, but the mutual acceptance of data. And all it does is tell you the study's been performed to an appropriate method in a suitable place. You still need to look at the results. But we should also remember the obvious. When we're talking about food chemicals, generally, we're not dealing with very toxic chemicals. Apart from pesticides and veterinary medicines, most food chemicals aren't designed to be biologically active. You look at a lot of studies with no findings because, remember, studies are a compromise between group sizes and doses for demonstrating safety to the required standard and animal welfare and other concerns. And just because the studies are done in isolation does not mean the evaluation is. You're always asking, are there differences in their design or conduct? And if so, why? And are their results consistent with other studies on the same compound or with what we know generally? As I say, integrating the evidence is key, and preferably answering that last question, do we know anything about the mode of action of this compound? And the data sources. One complication with food chemicals is we very rarely have human studies because of ethical and other reasons. But really, we're trying to protect humans. And unless we can demonstrate that our animal studies and the in vitro studies are reliable models for the humans, we cannot demonstrate safety for humans. But remember, they're models which may or may not reflect reality. Because once we have the critical effect and the benchmark dose and effect level, we take that into our risk assessment, taking the benchmark dose and effect level and applying uncertainty factors because of the fact that we're only testing small numbers of animals and that humans and animals may vary in their sensitivity to derive a safe dose, which will be below and often orders of magnitude below that no effect level in the animal. So we turn to the bit I spoke about, the crystal ball. What challenges do the future hold for toxicological testing of food chemicals? Well, I think there are going to be continuing testing from limitations on animal test methodology for welfare reasons. The limit tests that the maximum amount of compound given to an animal allowed today are significantly lower than they were in the past. And some of the ADIs that we have for existing compounds, you can never get today if we continue to use the uncertainty factors we have always used. And just because we can measure something and it changes, does it always mean it's adverse? Many of our concepts were based on overt apical effects, and we need to think about a new normal and think about what that might be. But we also need to think about applying new approach methodologies and omics. And we have to ask the question, how much validation do we need? We have many roadmaps, but maybe it's time we were radical and just used it. But we'll always have that question. What do we do about the unknown unknowns, as Donald Rumsfeld said? But before I depress you, we've been regulating food chemicals now for over half a century. And it's evolved our assessment as scientific knowledge has increased and improved and continues to do so in future. Thank you. And are there any questions?